So we left off with talking about monohybrid crosses and how Mendel was able to prevent self-fertilization, and this really allowed him to study the very specific inheritance patterns in different parent plants that had very specific phenotypes. So Mendel used a monohybrid cross as his very first experiment, where he actually looked at the phenotype of seed shape. So the two phenotypes he was looking at was round seeds versus wrinkled seeds. So he first crossed these two homozygous varieties of peas, and he found out that they were homozygous because he took 2 years to select the parent lines. And so, as you can see in the figure over here to your right, we've got a round seed, and that plant was crossed with a plant that produced a wrinkled seed. And in that F1, or the first filal generation, what ended up happening was that all the F1 seeds ended up being round. So Mendel really allowed these plants to grow. Um, grow into a fully mature plant, and then he allowed them to self-fertilize. He didn't control the fertilization process. And that F2 generation, or the next generation, um, ended up with a very unique set of ratios. So he actually did this study about 60 times, and he found that there was about 5,400 round seed phenotypes and about 1,800 wrinkled seed phenotypes. So when he did the data analysis on that in very simple longhand, no calculator ways, he found that about 3/4 of the offspring had the phenotype of round, and 1/4 had the phenotype of wrinkled. So this really resulted in a 3-to-1 ratio. Mendel's second experiment, he actually did a reciprocal cross, which means that he took, in the first study, he one plant, the male was round and the female was wrinkled, and he did the reciprocal cross in the second study, where the female had a round seed and the male had a wrinkled seed. And so he crossed those individuals, and then ended up finding the same exact results. So the F1 generation ended up producing 100% of the F1 seeds that were round. And then after subsequent self fertilization and studies, he found the same 3-to-1 ratio, regardless of what the original parent breeds ended up being. So the conclusion that he ended up determining was that the traits actually do not blend. So they never come together, and they never really start or blend and are partially expressed. He also found that although the F1 plants displayed the phenotype of one parent, both of the traits were actually passed to the F2 generation and resulted in that 3-to-1 ratio. So he did both the first experiment and the second experiment a total of about 60 times and got the same result every single time. So, some of the other conclusions that this led Mendel to come to was, first of all, he determined that the F1 generation must inherit both uh, or must inherit the genetic factors from both the male and the female parents, because they transmit both of those factors to the F2 generation. So he then went on to conclude that each plant actually will possess two genetic factors. And this is where the concept of the allele was born. So the allele is still coding for the same trait, there's just different versions of that trait. So then Mendel really started to look at how transmission happens. So how these traits are passed from one generation to the next. And we know, because you guys have already been through molecular genetics, that the organism behind that is DNA. But at this point in time, they did not even know that DNA existed. So they were simply studying inheritance patterns and relating that back to certain traits. So Mendel posed the question, how does this transmission happen? And what he ended up determining was that the two alleles are going to end up separating when the gametes are formed, and then one allele is going to be inherited into each gamete. And so when fertilization occurs, the two alleles actually come together and create the genotype, which in turn produce the phenotype or the physical trait. So this concept led him to determine the relationship that some traits ended up actually occurring in a greater percentage compared to other traits, and that led to him determining that some traits or some genotypes were actually dominant over the other genotype, which he termed recessive. So we know that we have this dominant and recessive phenotype and that is going to be present. And we now know that those, even when we have a heterozygote genotype, so like a big R and a little r, the phenotype ends up being round. So that indicates that the big R genotype is dominant, the little r recessive genotype. So, as we look at how these alleles are actually flowing from one generation to the next, if we look at our parent seeds, so we've got our round seeds, which are going to have a genotype of big R, they're homozygous, and we're going to cross that to a homozygous recessive genotype, or the wrinkled seeds, which is little r, little r. In the F1 generation, as gametes are forming, the two alleles are going in the plant are going to separate when the gametes are formed, so that there's only the chance of a big R coming from this parent and a little r coming from this parent during the fertilization process. So once fertilization occurs, the gametes are going to fuse to produce a heterozygous genotype in the F1 generation that we see here. And that's going to produce round seeds because the round genotype coded for by the big R is dominant compared to the wrinkled. So then Mendel went on to study the next generation, the F2 generation, and what he allowed to happen was all the F1 progeny to self fertilize. And so in the F2 generation, what he ended up with was these fractions. So he had about a quarter of the seeds actually being round and with the big R genotype, and then half of the progeny actually being heterozygous and having a big R, little r genotype. And then finally, a quarter of the leftover peas actually had a different phenotype. They had the wrinkled phenotype and had the homozygous recessive genotype of little r, little r. So then, when the F2 generation was allowed to self fertilize, we see that certain alleles are going to be able to be passed on. So, a big R is going to come from the homozygous dominant genotype. Either a big R or a little r is going to come from the heterozygous phenotypes. And then a little r is going to be the only allele that can be passed on from the homozygous recessive genotype. So when self fertilization took place and he looked at the progeny in the F3 generation, what he ended up finding was that there was a large percentage of individuals that still were round in phenotype, and they could either have the homozygous round genotype big R, or they could have the round phenotype but have a heterozygous genotype of big R, little r. And finally, the rest of them were going to be the homozygous recessive phenotype little r, little r. So this really led to the concept that Mendel termed as his first law, which was the principle of segregation. And we talked about this in chapter 2. This is just a really good review for you. So the principle of segregation states that alleles will separate or segregate in such a way that each gamete is equally likely to contain either member of the pair of heredity determinants. And so those are going to be the alleles. And so Mendel's first law can be visualized with this diagram here, where we've got um, the the chromosome coming from mom is red, and the chromosome coming from dad is blue. And on that chromosome, we've got a big R allele for seed type, and from mom, and on the allele from dad, we've got the little r phenotype from, or the big R genotype for seed shape. So as we are going through, as those two alleles um, are on homologous chromosomes, they are going to go through the replication process, which happens in the S phase of meiosis. And then in prophase 1 of meiosis, we're going to see crossing over either take place, like over here to the right, or not take place, like over here to the left. So both of those possibilities exist. Regardless, after the prophase 1 of meiosis 1 process, in anaphase, we're going to get the homologous chromosomes are going to separate, whether they've crossed over or not. And then just realize that if no crossing over takes place, the two chromatids of each chromosome are going to segregate in anaphase 2, and they're going to be identical. So we've got our big R genotype that are going to segregate out in anaphase 2, and then we've got our homozygous recessive genotypes that are going to filter out in anaphase 2 as well. If crossing over does take place, then the two chromatids are no longer identical, because they have one genotype and another genotype on the homologous pair of chromosomes, and that happened because of the crossing over. So, because they're no longer identical, the different alleles are then going to segregate um, and have different genotypes in anaphase 2. So that whole concept really led Mendel to the chromosomal theory of inheritance, which is kind of like a sub principle of his first principle. But this is really where genes are located on chromosomes, and as they segregate into cells, they're going to do so in meiosis. All right, so we're just going to start talking a little bit about how to predict the outcomes of genetic crosses. And one of the ways that Mendel actually used quite often in his experiments was by conducting a back cross. So a back cross is where you are going to cross an F1 parent or genotype to, or an F1 genotype to either one of the parent genotypes. So you're actually crossing an F1 generation product back to the P generation. And what a back cross will actually tell us, and really told Mendel, was that the tall phenotype was dominant over the short phenotype. And so when we look at back crosses, we can actually use Punnett squares to filter out how these alleles are actually segregating and the probability that they're going to segregate into a certain configuration. All right, so we're going to come back and talk about more prediction of outcome of genetic crosses in the next section.